Hi, I'm now going to talk about paper 51, May-June 2015. Question 1. The information on the label of a bottle of one brand of cleaning solutions for contact lenses states that it contains enzyme which remove protein from contact lenses. And you should know contact lenses are meant to provide crystal clear visions. But soon after a while, the contact lenses will become hazy, and the reason is because some of the protein that is embedded inside the contact lens for the purpose to make the lenses to be more comfortable while wearing it, over the time this protein will start to leak out and they will start to adhere to the contact lenses and it will cause the contact lenses to become hazy. So you will need a cleaning solutions that contain protease enzyme that help to remove the protein from the contact lens. The instructions are the following. First step, cleaning solution should be poured into cleaning pot provided up to mark level. Let's say this is the cleaning pot. They are provided with a mark level. You pour the cleaning solutions up to this mark level. Second step, contact lenses should be left in the cleaning solutions for a minimum for 4 hours. Now this is a very long time. Put the contact lens inside, incubate inside for 4 hours for the enzyme to totally denature the protein on the contact lens. And the last step, before using the lenses, they should be washed in a sterile saline solutions that containing sodium chloride. So you can't straight away take this insert into eye because these solutions containing enzyme. So you need to wash it using provided saline solutions. The content of the solutions are, now this is your cleaning solutions. Inside these cleaning solutions, they have 3 items. The first item is the most important item, the enzyme. And this is a type of protease enzyme, meaning that it is going to break down the protein. This enzyme name is called as subtilisin A. They also give you some of the characteristic of this enzyme. It says that the optimum temperature of this enzyme is 60 degrees Celsius. Optimum pH is in a range of 7 to 7.5. Slightly neutral. Buffer saline, the other things that's inside these cleaning solutions is buffer saline. Now buffer is always act of a balance of ions and balance of pH. Now in this case, the buffer saline is the one that mimic the physiological solutions as the body fluids near the eye area. You also have disodium EDTA inside the cleaning solutions that act as a preservative. Now this one is to disinfect the lenses to prevent any microorganism inside and so that they can safely insert it into eyes. A student was asked to find out the actual concentrations of protease present in the contact lens cleaning solutions. So this protease enzyme is unknown concentrations. The students are required to find out actual concentrations. And this you have did it the similar test in the lab before, where you will need to carry out serial dilutions and then calculate the rate of the enzymatic activity. Draw a graph. From the graph, you can draw a line and estimate the actual concentrations of the unknown enzyme solutions. Next sentence here. The student found a website that gave the concentrations of subtilisin A in different brands of contact lens cleaning solutions. This range between 20 microgram per centimeter cube and 100 microgram per centimeter cube. Now this one should be your independent variable. Now they already tell you the concentrations of the marketed cleaning solutions, concentration enzyme is between this and this. So meaning that these actual concentrations, the unknown concentrations of the proteus should be fall within this range. So you should do the dilutions within 100 microgram per centimeter cube to 20 microgram per centimeter cube. The students stimulated a dirty contact lens using protein gelatin and a thin transparent plastic sheet. One side of plastic sheet was dipped into melted gelatin containing a dye. The gelatin was then allowed to set. This is figure 1.1. This is a simulated contact lens, meaning you do a model to mimic the contact lens, so simulated contact lens. And contact lens are mimicked by this plastic sheet. So this represent the contact lens. And the protein on the contact lens are by this gelatin protein. So you are going to put this one inside a cleaning solutions. And this, they cut it into 10 millimeter times with 10 millimeter square. So this is the one that mimic dirty contact lens. The student tested the activity of subtilisin A. They want to look into how this enzyme functions, the activity of enzyme, by recording the time taken for the colored gelatin to be removed, leaving the transparent plastic sheet. Time taken for colored gelatin removed. This is what student recorded, so you know this is your dependent variable. The results for the color and so they want to record the times when the plastic sheet become transparent again, where all the gelatin is being removed or broken down. Figure 1.2 shows apparatus the student use. You can see this is a simulated contact lens suspended in a cleaning solution. So this is the cleaning solutions. This is the cleaning pots and line indicating 10 centimeter cube on the pot. So this one is your control variable that fixes to 10 centimeter cube of the cleaning solutions. And when you put it inside, they will check on the time when if this contact lens become clear again. Table 1.1 shows the result of trials to find the time taken for the colored gelatin to be removed by the cleaning solutions at 35 degrees Celsius. Now they fix it to 35 degrees Celsius. Again, this is your control variable. The student ran 3 trials, trial 1, 2, 3, and they got these results. In trial 1, 35 minutes is needed to remove the gelatin. Trial 2, 50 minutes is required to remove colored gelatin. Trial 3, 40 minutes is required to remove colored gelatin. You can see this data is not consistent. The student noticed that there was a large variations in the results. This may be because it was difficult to know when to stop the stopwatch. Now you are looking into when colored gelatin is being removed and you are looking at the color. So suggest two problems in deciding when to stop the stopwatch is in these investigations. This one, they ask you to identify the source of error. So the problem why there is a large variation in these results is because very difficult to identify endpoint. Now you may sometimes, the student might sometimes stop the stopwatch earlier, or they might stop the stopwatch too late after the endpoint. So this is a type of random error. You have two types of error. One is systematic, another one is random. In this case, it's considered under random error because sometimes it will be less time, another times will be more time recorded because it is very difficult to stop the timer precisely. Difficult to identify endpoint because in this case the gelatin is digested gradually, and also because the dye color. The dye colors the cleaning solutions, causing solution becomes cloudy. So the solutions might not be enough, might not be clear enough. Second, the student make a stock cleaning solutions containing 1 milligram per centimeter cube of subtilisin A, disodium EDTA, and a buffer saline solutions. So this is the stock cleaning solutions. They contain these three items. And the disodium EDTA and buffered saline solutions were the same concentrations as used in trial. This solution was used to make a range of concentrations that could be used to find the concentrations of subtilisin A in the contact lens cleaning solutions. So they ask you to make a range of concentrations that allow you to estimate. You have know about the estimations that these concentrations must be fall between 100 microgram centimeter cube to 20 microgram per centimeter cube. So you need to do these dilutions within that range. Describe how the student could dilute the stock solutions to make a range of concentrations to use in their investigations. The students decided to make 50 centimeter cube of each concentration. This is V2. So for this, first thing you must know you want to prepare 100 micrograms per centimeter cube. So the stock solutions is way too concentrated. You need to dilute the stock concentrations. Dilute stock concentrations or stock solutions, that is 1 milligram per centimeter cube by a factor of 10 to give 100 microgram per centimeter cube solutions. And how you're going to prepare this is to make 100 microgram per centimeter cube solutions mix. Now you need to decide mix how many of the stock solutions. So you should do with this M1V1 and M2V2. So you have M1, V1, M2, V2. M1 is your stock solutions. In this case, you change into the same unit number. 1 milligram per centimeter cube equals to 1000 microgram per centimeter cube. So this is M1. So this is the original concentrations. You don't know what is the volume that you want to take it for dilutions. M2 is the diluted concentrations you want to make into 100 microgram per centimeter cube. V2 already provided to you in the question. It is 50 centimeter cube. So V1 should be 5 centimeter cube. So you need to mix 5 centimeter cube. And if want to top up to 50 centimeter cube of the total volume. So you need to mix this 5 centimeter cube of stock solutions. Stock solutions is this is 1 milligram per centimeter cube. Mix this with 45 centimeter cube of saline solutions. You want to make the total of the volume here to be 50. So you need to mix with another 45 centimeter cube of, you cannot write distilled water because in this case, their buffer is using saline solutions. You can just write EDTA and buffered saline solutions. Part 1, now you already got this 100 microgram per centimeter cube, so you can start to prepare the range of concentrations. Prepare 100 microgram centimeter per centimeter cube. The next one, you can put it 80, 60, 40, 20, so at least minimum 5 dilutions concentrations. And you need to mention how you're going to prepare it. You can prepare it either by simple dilutions or serial dilutions. And you can give an example. For example, how you're going to dilute to make 80 microgram per centimeter cube. So to make 80 microgram per centimeter cube, mix for this one you should also calculate. For example, for to make 80, and again using this formula, M1V1, M2V2. M1 is your original stock solutions here. So you are going to dilute from 100 microgram per centimeter cube. So you put 100 inside here. V1 is the one that you want to find out. You want to find out what is the volume that you should take from these stock solutions to make dilutions. You want to make the dilutions into 80. So 80 is the diluted concentrations. Final volume is you want to top up to 50. So V1 should be equals to 40. So you should mix 40 centimeter cube of 100 microgram per centimeter cube of the cleaning solutions, or you can write enzyme solutions. And then you need to top up the total of 50 centimeter cube. Mix this with 10 centimeter cube of saline solutions. But in this case, marks will be given if you talk about the range that is within 120 micrograms centimeter per centimeter cube. So you get 1 mark. What is the method that you use? Whether it's serial dilutions or simple dilutions, you will get the second marks. And next one is if you say you dilute the original stock solutions to give this. So this one you will get 1 mark. Design how you're going to make the dilutions. If you get it correctly and your total volume add up is 50 centimeter cube, you will get another marks. State the solutions that students should be used as control. Give a reasons for your answer. Now control is where you need to use to ensure that your result of the experiment is due to the independent variable but not other factor. Is due to your enzyme but not due to other factor like the factor of the saline solutions might containing or contaminated with other protease enzyme. So the control here, control solutions for this case of the enzyme experiment is always boiled enzyme solutions, so that the enzyme inside it is not functions. So in this case, these solutions is supposed to get negative results. There are no degradations of the protein. There are no changes of the color of the gelatin. Or another thing ways is if you are not going for the boiled enzyme solutions, uh, just don't put the solutions with enzyme. So make sure the solutions is without enzyme. The volume of the solution should be the same. It's always 10 centimeter cube, but it is without enzyme. Now the reasons why you need to include these boiled enzyme solutions or include the control is to show that other components of the cleaning solutions. Other components of the cleaning solutions including the EDTA, including the buffer saline, do not digest the saline, do not digest the gelatin. Or you can just write to show that the enzyme is the one that digests protein. So in this case, the control should show totally negative results, meaning that they are very not a long time or infinite time. You just put that there are no degradations of the protein at all. And this also can prove that there are no other enzyme that is present inside the solutions. So it also prove that the result of you getting here is purely due to the protein subtilisin A, not other enzyme that is present in the solutions. Next one, the student used the apparatus shown in figure 1.2 to investigate the effect of different concentrations of subtilisin A on the rate of removal of colored gelatin from the simulated lenses at 35 degrees Celsius. What is the independent and dependent variables? Now, the independent variables is the variable in an experiment that is manipulated or changed. And dependent variable is a variable in an experiment that is measured. So in this case, what is the things that is changed in the experiment? What is the variable that changed? That will be the concentrations of subtilisin A. And dependent, you are checking for the time for disappearance of gelatin. Describe a method students could use to find the effect of different concentrations of subtilisin A on the removal of protein from the stimulated lenses. Your method should be detailed enough for other persons to follow and should not repeat any details from B1 of how to dilute stock cleaning solutions. So no need to put how to dilute. You can just exclude this one. In this case, describe a method. You should always put it independent variable. You should describe how you're going to do the dependent variable, how you're going to measure it, and also you need to mention about control variable, the safety of the experiment, and the reliability of the experiment. So we will now start to look into what is the independent variable in this experiment. So independent variable, it is where you prepare all those solutions in different concentrations. But the question here tell you that don't mention or don't repeat how to dilute the cleaning solution. So you can just straight away, once you already have these stock cleaning solutions at different concentrations, then you need to use apparatus to measure and pour it into the pots, into the cleaning pots. So the first one is you can use any equipment, for example, string or measuring cylinder or graduated pipette. So you can talk other things like string or measuring cylinder. And now you're going to measure the diluted stock. Now the stock solutions is you need to measure how many volume, and it is given in here in the diagram figure 1.2. So you're going to measure 10 centimeter cube of the clean solutions. To measure 10 centimeter cube of cleaning solutions. And fill in each pot. Now, another things that you should talk about is after you put into each pot, you need to make sure all the enzymes should be at 35 degrees Celsius. You need to talk about all these control variables. So you need to make sure that the enzyme, you need to incubate the subtilisin solutions for 5 minutes in 35 degrees Celsius temperature controlled water bath. before adding the stimulated contact lens. And you can talk about the dependent variable. You're going to measure the endpoint, the color changes. So number 3, always mentions about the apparatus that can help you to measure. Use stopwatch to measure endpoint. The endpoint will be the time for the colored gelatin to be removed by the cleaning solutions at 35 degrees Celsius. Next one, you can talk about control variable. Now control variable is you need to, what else do you need to standardize? You need to standardize the thickness of colored gelatin or the mass of the gelatin for each experiment, and you need to use buffer to keep pH constant. Number 6, use same size of contact lens for each experiment. Number 7, other than this, you can talk about the safety of this experiment. So this is low risk experiment, risk assessment. What is the possible risk? Enzyme might cause skin irritations or skin allergy. So wear glove during experiment. Number 8, you can talk how to increase the reliability of the experiment. You can increase the reliability by repeat the experiment 3 times and calculate mean value. So you can look inside here. Now the first one here is the independent variable. What is the volume, concentrations of the testing solutions, and this one should be the control variable, where you standardize the temperature. You can get the temperature from the question. They say it must be in the 35 degrees Celsius. Next one, you should talk about how you can get your dependent variable. What is the measurement? How you can measure by using stopwatch. And from here to here, this is the control variable where you standardize the temperature, the thickness of the color gelatin, standardize the pH, how by using buffer, standardize the contact lens. and all the contact length should be, if you refer to the diagram before, it is 1 millimeter times 1 millimeter square. And then you should talk about the risk assessment of this experiment. So this is the low risk experiment, or the safety of this experiment, and then talk about the reliability, how to increase the reliability of this experiment. Question D1. The student plotted the graph of the results. Complete figure 1.3 by adding labels for the axes, including units, and by showing the expected shapes of curve. So what is your x-axis? Each x-axis should be the independent variable. So here should be always independent variable. Y-axis is always your dependent variable. So the independent variable here is concentrations of subdilution A. And the y-axis is the dependent variable, is the time for disappearance of gelatin. And draw the graph here. Now, if you have more concentrations of the enzyme, meaning that you will require less time for the disappearance of gelatin. It will disappear faster. So it should be look something like this. Now, before forget, make sure that your x and y axis contain unit. So in this case, the time could be either second or minute. These concentrations of subdilution A should be, if you look into your question B1, if you look into your question before, they said that there is a range between 20 microgram centimeter per centimeter cube. So the unit should be microgram per centimeter cube. Marks will be given for correct label of x and y axis, and then with a correct unit, so 2 marks here, with an appropriate graph. When you have increased concentrations of enzyme, decrease the time for disappearance, meaning require less time. Number 2, describe how the student would use this graph to find the actual concentrations of subdilution A in the contact lens cleaning solutions. First one, find the time. So they need to carry an experiment to check the time for the gelatin to disappear. And second, draw a line on y-axis and read off the concentrations of subdilution A on x-axis. So after you perform the experiment, you got the time for the gelatin to disappear of the unknown concentrations. So you can just draw, let's say this is 10 seconds. So you just draw a straight line from the y-axis, and you read off the concentrations on the x-axis. So you got the concentrations of subdilution A in the cleaning solutions.